So now, if you now look at this, you will understand that in this case, the concentration of sodium chloride and water molecules are the same in the cell and outside the cell. And that is why no movement. So they are at equilibrium, they are equal. Then now, in the case of hypotonic solution, you see in the cell, the concentration of sodium chloride is 20%, and outside the cell, that is in the environment, we have 10% of sodium chloride. And then for water, we have 80% in the cell, and we have 90% of water in the environment. So you see, it is not the same. So now what will happen? What is the direction of the water movement? Of course, the water movement will get into the cell so that at least the concentration or the amount of water in the cell will be increased or will increase. That is why you have this arrow pointing into the cell, because the water outside the environment is higher than the amount of water inside the cell. So this is the direction of the movement. And then in the case of hypertonic solution, you see the concentration of salts, the concentration of salt is 5%, and the concentration in the cell, and then the concentration of salt in the environment is 15%. The concentration of water or the amount of water inside the cell is 95% and 85% in the environment. So therefore, the direction of the movement is outside the cell, that is through the environment, so that at least the concentration of water will now increase for the equilibrium to be maintained. That is in the case of hypertonic solution. So now, a more look at this thing, the conditions. External solution is hypotonic to cytosol. That is so okay. Now the direction of osmosis. Now these are the conditions. So in the case of the hypotonic solution, the external environment or the external solution is hypotonic to cytosol. So therefore, the net movement of water is into the cell. So that is why if you look at this, this is a typical example of cell membrane. So you have molecules of water are getting into the cell, and this is because in the environment, the concentration of water is higher than that of the concentration of water into the environment. So that is why more water is needed to get into the cell in order to maintain equilibrium system. Then in the case of hypertonic solution, it means that the concentration of water outside the environment is higher. So that is why you need, outside the environment is lower, and the concentration inside the environment is higher. So that is why you need to move out the water outside the environment for the concentration of water to increase in order to maintain the equilibrium system. Then in the case of the isotonic solution, then the concentration of water are the same. So therefore, there is no non-movement. It means that, of course, when you said non-movement, it means that it's not that nothing is moving. No, it's moving, but the concentration at the cell is the movement is not only in water, sorry, into the cell or outside the environment. Both is actually taking place. So that is in the case of this. You see, in the case of isotonic solution, no net movement of water, that is equal amount entering and leaving. Equal amount of water is getting into the cell and leaving the cell. So that is why you have this double arrow. This one is indicating that it is getting in, and this one is indicating that it is getting out. And then in the case of a hypotonic solution, we have cytolysis. Cyto means cell, lysis means breaking down. So there is a chances that the water, or sorry, the cell will break down in the case of cytolysis, sorry, in the case of hypotonic solution. Then for the hypotonic solution, we have plasmolysis. That is the shrinkage of cell. Cytolysis is the bursting of the cell. The cell will burst. So cytolysis, cell placed in distilled water, and this is a red blood cell and is placed in distilled water. So the cell wall or the cell is swell and bursts. That is in the case of cytolysis. Then cell placed in concentrated salt solution, where the concentration of salt is very high, then the cells will shrink. That is plasmolysis, the shrinkage of cell. So that is in the case of cytolysis and plasmolysis. So when you actually place your cell in a distilled water, then the cell will swell. The cell will swell and boost. And then when you put your red blood cell in the concentrated salt, it will shrink. This is osmosis in red blood cell in both hypotonic, isotonic, and this, okay? So what happened to red blood cells? So red blood cells, we have water. So when you have hypertonic solution, the water is actually going out, or it's leaving the cell. And then in the case of the isotonic solution, the water is leaving and also getting into the cell. In the case of hypotonic solution, the water is getting into the cell. So you should understand this. So it means that now solute concentration in extracellular space, it means that in the environment, the concentration of salt is high. And then here, there is no difference in terms of the concentration of salt in and out of the cell. But in this case, the concentration of salt in the case of hypotonic solution. So this is the structure of the structure and the function. So the drawing below show the appearance of a red blood cell and a plant cell in isotonic, hypertonic, and hypertonic environment. So this is just, it's like a kind of an assignment. So how can we just label this? A is what? B is what? Let me just get this from you. Who can tell me A is what? In the case of red blood cell, A is what?
A is isotonic.
Then B.
Isotonic, sir. B is hypertonic.
Hypertonic.
Then what of C?
C is hypotonic.
Okay. So what of in terms of the, look at the plant cell?
Plant cell, D is isotonic.
Okay. Then what of F?
Isotonic.
What of F?
F.
F is
But sir, E.
So you sit down. Actually, I'm going to send you this. So you see, look at it. D is hypertonic. This one is isotonic, and this one is hypotonic. So now, ladies and gentlemen, we are now going to look at the.
The slide again.
Sir,
It's time.
no enter at all.
Slide. The labeling, please.
Okay.
Thank you, sir.
That explanation is not really clear.
Okay, this is a plant cell, and this is a mitochondria. This is what? A mitochondria. And of course, remember in a cell, we have cell wall. We have cell wall. So now, look at this. This is the natural structure of the cell, in this case of isotonic. That is the natural structure. So look at it. But in the case of hypotonic, remember what happened to the cell when it is hypotonic? Is it plasmolysis or cytolysis?
cytolysis, sir.
Cytolysis. That is a breakage of a cell. But because of the nature of the cell wall, it might not necessarily to actually affect the integrity of the cell. Do you understand?
It's not clear. I don't understand it.
Okay, because of time factor, we'll discuss this maybe on WhatsApp. We'll discuss this on WhatsApp.
Okay.
Okay.
Yes, sir. Thank you.
I have a question.
Okay.
Yeah, it's regarding the